My name is Kato from Kyoto University, and I'm going to chair a session this morning. And the talks, well, there are 3 talks in the session, and the talks this morning focus on sort of a basic biological phenomenon. And the first speaker is Professor Kunihiko Kaneko from University of Tokyo, and his title is Consistency Principle in Biological Dynamical System, and so on. He's going to explain. So, Professor Kaneko, please start.
Yeah, thank you very much. So today I'd like to talk about some general properties of biological systems. And indeed, I'm proposing the study of complex systems biology. And in the complex systems biology, we try to understand some universal features in biological dynamical systems. And in the complex system, I mean the system that has some kind of mutual dependence or a mutual relationship or feedback between each part and the whole system. So, and the guiding principle today is that to see some consistency between different levels. For example, in a cell, you have several molecules. And so these molecules have to reproduce for cell reproduction. So molecule replication and cell reproduction. And so it's a different level. But to keep some kind of recursive production, they need to have some kind of consistency between levels. And in the statistical physics, we often consider each molecule and ensemble of molecules, and so it's just from the direction from this part to this part. But in the biological system, nature of each part is often determined by the whole system. For example, in a different level, cell and multicellular organism. And so ensemble of cells, and the nature of cells is somehow determined by other distribution of other cells. So it's kind of mutual dependence. And also, maybe yesterday or the other 2 days before, we had a discussion of genetic and this phenotype. And so in this case, gene is somewhat influenced by this phenotype also through evolution. So I'm working on these kind of studies in the different levels. So for the problem of reproduction, evolution, adaptation, and development. And so today I think I mostly focus on this evolution. That is somehow consistency between genetic change and phenotype change or phenotype fluctuation. So this consistency and some general consequence from that consistency. But maybe I talk a little bit, start from this consistency between cell reproduction and molecule replication and some universal statistical laws derived from this. And so maybe if I have time, I slightly mention about adaptation and development also. So the underlying biological motivation for this evolution study is that somehow we have some kind of feeling that biological plasticity, phenotype fluctuation, evolvability, and these type of words. And they are sometimes very vague. But I want to try to more quantitatively define these in terms of phenotype fluctuations. And so by that, we try to make a kind of link between development and evolution, and to discuss evolution of robustness. So in terms of physics, maybe, so which type of system is selected is more important. So in the physics studies, we start from some kind of system and study how the evolution dynamics lead to some behavior. But here, through the evolution, which type of system is selected is more important. So I discussed this here. So but I start from this problem. And so the question is very naive. So how recursive production of a cell is sustained? So we consider a very prototype or primitive cell from the beginning. And so each cell has a complex catalytic reaction network with diversity of chemicals. And then each molecules are somewhat reproduced and then grow and divide. But considering the diversity of chemicals and these fluctuations of each number, it may be quite difficult to keep recursive production. So I start to consider some conditions for this. And so in the beginning, I start from a very simple kind of poor physics model of a cell. And so this is some kind of catalytic reaction network. So you have maybe 1,000 chemical species. And this is transformed to some other molecules and transformed to some other molecules through this catalytic reaction, but catalyzed by some other molecules. And so this is basically catalytic reaction. So this type of reaction. This is changed to this with the aid of this. So I is changed to K with the aid of J. And so here, so of course, for a cell to grow, we need some kind of nutrients. So nutrients are flowing in. And then if this reaction works well, this is transformed to some others, and this cell may grow. So the number of molecules there grows. And so I assume that when the total number of molecules is larger than some threshold, then it's just divided into two. And in that case, so just stochastically, the number of each molecule is distributed. So there are many sources of fluctuations. And so here we take very simply just random catalytic reaction networks. Of course, biological network is far from random network, but we start from just randomly wired network and study some kind of general properties there. And so basically, the model parameter is that some kind of diffusion coefficient, so the speed of flow of this nutrient to here, and so the number of molecules there, total n, and the number of molecule species. And so we did this simulation. So we can do this by using ordinary differential equation like this, but maybe we mostly use just simply stochastic simulation. So just choose two molecules and collide, and if it reacts, it's changed. So, and the results here. And so if we increase T, then the speed of flowing nutrients increases. So it's quite natural that growth speed, that's the inverse of the division time of this cell, increases with this D. So D and this graph data grows. But we found that if D is larger than some threshold, then this cell can no longer continue to divide. The reason for this is as follows. If D is too much, and then this nutrient comes very fast, and there is these other catalytic reactions cannot follow. So after some time, this cell becomes just nutrients, and there are no, for example, no catalysts here. Then the reaction is stuck here, and then so finally it goes to equilibrium with the outside, and there is no more flow. So for this cell to grow, so this reaction process has to accompany with somehow, so keep synchrony with this diffusion speed and this reaction process. And so if this is too much, it stops there. So in some sense, we need some kind of balance between this reaction process and this flow, nutrient flow. And so this balance is broken down here. So, and actually, we checked so similarity of this cell. So the similarity is defined from how the cell, daughter cell and mother cell are similar. Similar means that the chemical compositions are similar. So here, for example, you have 1,000 chemicals, and then this state is represented by a kind of vector in this 1,000-dimensional space. So if this mother cell and daughter cell is identical in composition, then it's 1. So it's in a product. But if it's different, it's 0. So, and we define similarity. So anyway, this is how the chemical compositions are similar between mother and daughter. And so this increases like this, and this is 1. So here, it's very close to 1. So that means around here, the cell can grow efficiently and produce almost recursive production. And actually, the reason that cell can produce almost the same chemical composition is that it produces some kind of structure in the through the catalytic reaction. And so I checked some kind of statistical behavior around here. So here we plot the, so actually in this model, so there are many chemical species, 1,000 chemical species. And some are very abundant and some are not so much abundant. So for example, chemical species 1 has 3 and 300 and 2 is 8,000 or something like that. And we just put the ranking and the number. So in this case, this is the ranking 1 and this is the second ranking. So we put ranking and this. So of course, then this is a decreasing function, this ranking abundances. But here, by putting this log scale, we observe always this -1 exponent at this recursive production state. So that means average number is inversely proportional to the ranking. And so now, as this seems to be very universal, we checked if this is true at the present cell. And there are, these are 3, or we checked more than 100 cells. And always, this ranking and abundance obeys this law. And in this experiment, what we checked is by using microarray or some other techniques. And we checked the abundances of messenger RNA, or roughly abundances of protein. And then this ranking and abundances of each messenger RNA. And that obeys this structure. So this law that appears as a kind of general result, as a kind of recursive production of a cell, seems to be rather, universal, even in the present cell. And actually, we checked several models, and this result is quite universal. And we also did some kind of evolution experiment. So starting from some network and with different d values that does not satisfy this power law state. But then, through this evolution, this finally goes to this state by changing the network structure. And also, I said that in this, I choose random reaction network. But if we allow for the evolution of the network, so by changing the network structure so that this cell can grow more efficiently, then this embedded, this power law abundances, so this is abundances power law, is later embedded into the network structure. And this leads to, for example, a scale-free network. And we can explain this. And also, so some may wonder, so choose around this position, then if D is slightly changed, it dies. So that may not be so good. And so instead of choosing standard diffusion, as in the usual cell, we assume that we introduce, for example, kind of active transport. So this nutrient is transported into the cell, catalyzed by some molecules here. Then, by using such kind of active transport, then we can have a kind of self-adjustment of the state, because if the number of this nutrient is poor, too much, then, for example, if this molecule is catalyzing this here, and then if this is too much, this number decreases, so that this transport is suppressed, so that by it forms some kind of self-adjustment to of this behavior, and then in this case, so self-organization to this power law state. So that's an example that how this recursive production constraint leads to universal statistics, and that are true in the present cell. And next, we discuss about this fluctuation. The fluctuation, I mean, is that even I said that this is almost recursive. Of course, the number of these some chemical species is not identical between mother and dog. For example, if it divides in next cell, the number is slightly changed, and then the third division maybe is slightly changed. So there are fluctuations. And we check this fluctuation. And so here, so we take a histogram of this. So over many, many cells for some species. So given some chemical species and many cells, and this histogram. So this different color corresponds to different chemical species. And so this distribution is over cells. And so this looks like Gaussian distribution. But the point here is that this axis is log scale. And so this is always log normal distribution. So after taking log, this has a Gaussian normal distribution. And actually, this type of behavior is also observed in the present cell. So, for example, by putting a fluorescent protein in bacteria, and we can measure the abundances of such proteins through the fluorescence by using flow cyte method. And so that is the result here. So here we put 3 different proteins. And so this is log fluorescence, basically log abundance. And this obeys roughly, yeah. Anyway, after taking log, it is very close to normal distribution. So that means there is very, very large fluctuation. Even the cells have same genes. And actually, the reason for this log normal distribution, I think this is quite simple. Because basically, for this cell to grow, this has to be catalyzed by some other molecules. So basically, the number of molecules for given molecules is catalyzed by some others. And by that, it increases. So this equation, for example, is sort of transformed by this. But this number fluctuates. So in that sense, log n, log of the number, shows a Brownian motion. So roughly speaking. So that means, so log nx goes to Gaussian distribution. So we can elaborate this argument a little bit more by successive multiplication or something. But basically, the point is that it's a multiplication process, then the fluctuation is multiplied. Then it leads to log normal type distribution. And actually, we also, so this is the laboratory with the Yomos group. And we try to make an artificial replicating cell. And so starting from liposome, it's an oil membrane. And with that can grow and divide. And within this liposome, we make some kind of translation, DNA translation and protein synthesis. And that's all each part we succeed. But the difficulty here is how to synchronize these. So the process of this membrane division and this internal reaction. And so the synchronization between these is a difficult issue. And for that, maybe this consistency or some other argument is important. Now I come back to the problem of this fluctuation. Oh, okay, so this is the summary of this part. But anyway, we found that the phenotypic fluctuation is quite large. And so this is the fluctuation of isogenic organisms. So not due to the difference of genes. So we now I will discuss this relationship to evolution. And actually, selection is based on phenotype in this evolution. So phenotype is, for example, some cell activity, cell size, protein abundance, or fluorescence, or something like that. But in the standard evolutionary genetics, usually it's assumed that gene gives phenotype, and this is uniquely determined, basically. Then, instead of discussing of phenotype, we can discuss just genetic variation, so genetic distribution. So if gene corresponds directly to phenotype, we do not discuss explicitly about the phenotype distribution. Instead, we can discuss genetic distribution. Then, so we can have a field of evolutionary genetics instead of evolutionary phenotypics or something like that. So, but here, what we found is that this phenotype fluctuation is quite large. So we want to discuss its relevance to evolution. So to study that, instead of considering just the distribution of gene, we need the two variable distribution of phenotype and genotype. So we need to discuss phenotype and genotype two variable distribution. And now, so I want to talk a little bit with the experiment conducted together with Yomo's group. And so here, it's a kind of artificial selection experiment. So we have some kind of bacteria, and in this bacteria, we put some fluorescent proteins. But initially, this fluorescence is not so good, so rather bad. Very little fluorescence. And what we did is that make mutation to this bacteria for this gene for this fluorescence protein, and select such cells with higher fluorescence. So it's just a very standard artificial selection experiment. And so we repeat this. And then by doing this, so initially the fluorescence, this is fluorescence. So initial cells, the fluorescence average is here. But still, even for this same gene bacteria, so there are distribution. So phenotype is distributed, so fluorescence is distributed. And so, anyway, this is the initial. And then, from this we make mutants. So some have this, and some have this, and some have this, and some. And then choose the higher fluorescent one. So this is the one. So this is the first generation. Still, this has distribution. So this, from this same gene, this phenotype is distributed. And then from this, we make this and repeat this process. So by this procedure, of course, we have this fluorescence increases in these generations. But you may see that this evolution speed starts to decrease, and also this fluctuation starts to decrease. So somehow, phenotype fluctuation and this evolution speed may have some correlation. And if the phenotype fluctuation is larger, it has more evolvability or higher evolution speed. So we want to discuss this. And actually, in physics, so there is a famous theory about fluctuation and response. And that is called fluctuation dissipation relationship or theorem. And that was original version was proposed by Einstein about a century ago. And then later, so elaborated. And so here, it's basically this like this. So if you consider some kind of variable x, this x can be in some position or some velocity or some electric current or something like that, any variable. And by put some kind of environmental change, external change, so you put some kind of generalized force to this. And then this x variable changes, so it's shifting. So response ratio is basically the shift of x variable divided by force. But before this, even without putting force, there is some kind of fluctuation of x. So in the case of physics, this is thermal fluctuation. And so this fluctuation and this response ratio is proportional. And this proportional coefficient is generally determined by the temperature. And that is this theorem. So this can be proved for the thermal equilibrium state. But we can somehow generalize this argument. And so if you consider some kind of distribution of phenotype or anything else, and controlled by some parameter, and in this case, change. So you can consider this x is, for example, fluorescence, and A is gene. And so by changing A, of course, this distribution position changes. So it may go up, or it may slightly go up or something. So we want to discuss now, so the response ratio is that by changing A, how this X is changing. So this is the change of X, and divided by this A change. And what they say, well, if we generalize, is that this is proportional to fluctuation. And of course, this generalization cannot be perfect. So in the case of thermodynamics, we can, this is exact, but this is somewhat approximate. But anyway, we can have some kind of approximate relationship between these. So considering this relationship, and so we, yeah, this is mathematics. Yeah, we can do this. Yeah, and so we applied this idea. So change in GA, so this artificial selection experiment. So change in A is that change in G, and response is change of phenotype, and in this case, change increase of fluorescence index. So that means this part is increase of fluorescence divided by number of mutations. This basically this is evolution speed, and this is original isogenic phenotype fluctuation. And so we put this data, and actually, so this is evolution speed. So this is from this previous experiment. This is evolution speed per generation. And we need to, so in the previous argument, this should be divided by mutation rate. So this is mutation rate. But instead of doing this, we multiply this to here. So, and this is fluctuation. So this is fluctuation. So this is variance multiplied by mutation rate. Here. And so then, so the data is red one. So although the data are somewhat scattered, roughly, yeah, we can see some kind of proportional relationship here. And just for comparison, we put the data with naive expectation, just evolution speed is just proportional to mutation rate, and without any relationship with the isogenic phenotype fluctuation. And that data is this black one. And actually, in this experiment, we choose only two mutation rates, unfortunately. So there are just two values. But anyway, comparing this black data and red data, this red data is much more better. So this actually is. So that means at least phenotype fluctuation is somehow positive correlation to evolution speed. And so maybe some of you may not be satisfied with this scattered data. So we also checked this by using numerical simulation. And here we use the previous cell model that I explained. And the task here is that we choose some chemical, so here some X, yes, and increase this number so that this, so for example, this is kind of fluorescent protein, and try to increase the concentration of this by changing the network structure. So initially we have this, and we did this simulation, and we make a mutants. Mutants means that slightly changed, so you put some other path or some other or new catalyst or something like that with some mutation rate. So from each parent cell, we make mutants with different network, slightly different network, and choose that cell with this highest this value. So it's basically artificial selection type experiment, and continue this. And so we can discuss the evolution speed versus fluctuation. And fluctuation here I means that even for the same network, this number fluctuates. And the result here. So here this is evolution speed, and this is fluctuation. And starting from this, each course of the evolution, so with a different mutation. So that means evolution speed proportional to phenotypic fluctuation. In this case, it's more, yeah. Maybe you can agree that maybe I can put that straight line here. So far, this is fine. But here, there remains some mystery. So, okay, one mystery here is that, so we have seen phenotype fluctuation of isogenic organisms is proportional to evolution speed. But there is famous theorem in evolution that shows evolution speed is proportional to phenotypic fluctuation by genetic variation. And this is called the fundamental theorem of natural selection by Fisher. And this is somehow established. So the difference between these two are something like this. So for isogenic, so I discussed isogenic phenotypic fluctuation. The same gene organism, but this phenotype is distributed like this. And here I just call VIP, so V isogenic phenotypic variance. But in this case, gene is distributed. And for each gene, there is some given phenotype. And maybe it may be distributed, then in that case, the peak position here. So, this position. And so then, since gene is distributed, accordingly phenotype is distributed. And we can measure this variance. And this is distribution. So the standard theorem says that Vg is proportional to evolution speed. So if both are true, that means Vg should fall. But this is not trivial at all. Because Vg is that changing the network. So it's a different type of equation, different type of network. The VIP is same network, but there are some noise and fluctuation, and that is distributed. So it's a different concept. But so the question is that if there is somewhat relationship here. So to discuss, so I consider again this kind of consistency from genotype level to phenotype level. So here, gene is distributed, and from this, we have some kind of phenotype distribution, and then selection, and this feedback to genotype. So there is feedback process here. And so I consider just distribution of phenotype and genotype. And the assumption here is that through this evolution, this distribution has a single peak in this phenotype-genotype space like this. It looks very straightforward. So, assumption. So the assumption first is that we discuss phenotype-genotype distribution. We can consider this type of distribution. And then this distribution keeps single peak distribution through the evolution. So we do not consider like speciation or something like that. We consider here just robust gradual evolution, only that. So in that case of this. And then, so we consider this distribution around some point, and then from this, okay, maybe we can keep all these equations, but the result here by this calculation. First, VIP is larger than VG. And then, but VG increases with the mutation rate. This is due to genetic change. So if you have large, high mutation rate, VG increases. So that means if there is some too large mutation rate, VG increases, then this inequality is violated. So that means that there is some kind of critical mutation rate beyond which so some error catastrophe and distribution becomes flat. So that this is second point. And so if VG is roughly proportional to mutation rate, then that means by using this critical mutation rate, we can write down this, because at critical mutation rate, VG and VE equals, and VG increases with this mutation rate. So we can write down this. So that means with fixed mutation rate, this is proportional. So we have these three relationships, this and this and this. So this is somewhat confirmed experiment. So the 2 points here. This look a bit surprising because this means that phenotype fluctuation, isogenic phenotype fluctuation, should be larger some is due to the genetic change. So it's a bit surprising. So we need some confirmation. And so we did this previous reaction network cell model and measured VIP and VG. And the results are here. So VIP and VG. And so for fixed mutation rate, this both decreases through this both fluctuation decreases. And as we increase the mutation rate, it approaches this diagonal line. So that means always this is satisfied. And when this approaches this, what happens here is like this. So if we increase the mutation rate, at this critical mutation rate, this distribution becomes just flat. So before that, it has a nice single peak structure, but then around this critical mutation rate, this becomes flat. And then the evolution no longer progresses, because the evolution, we try to select some part of this good one to the next generation. But if this is just flat to this, we cannot, this part is almost zero. So this evolution no longer progresses. So all these three results, three propositions are satisfied in this model. And this is a bit surprising. We didn't assume any type of two variable distribution or anything like that here. And actually, the mutation occurs through the very high dimensional network structure, not to single scalar variable like this distribution function. So it's a bit surprising. And so there are several questions to this original theory. So we consider this distribution function. And here, we assume some kind of genetic phenotype correspondence because we consider two variables, genotype and phenotype. So that means what phenotype can vary is related to what genes can change. And that this argument is already discussed by the famous Waddington's concept, Waddington's genetic assimilation. So I'll come back to this later. And so the next question is that why Robustness occurs when this VG is larger than VIP? That means it's better to have phenotype noise. So it's quite counterintuitive. So to check this, we consider another gene network model. So here, we very simplified consider very simplified gene network. So basically, the gene is on or off state, and two variables. So this on state or off state. So xi is expression of gene, and this is in this model. So simply on state or off state, but it's continuous. So if this is in this model, if this is 1, this is on, and -1 is off, and that is related with other genes. So this is given by this Jij. So this Jij is unrelated to 0. And if one gene is activated, some other is +1. And it suppresses, it's -1. So the gene network structure determined by genetic sequence is given by this. And by that, so we have gene expression state. So x1 to some x4, for example, here, and this is +, -, +, or something like that. So we have some genes are on and some genes are off after some development of these dynamics. And so what I put through this evolution is that just a condition that some genes should be on after some time. So for example, starting from all of state, after some time, so for example, these genes should be on. So this is the condition. So if the number of on genes are larger, then it's much fitter. So we have a selection experiment like this. So choose this. So basically, we have, for example, the case of two variables. Starting from here, and the good one is that it goes to positive one, but some goes to negative or something. And so we try to choose such gene network structure to satisfy these dynamics. And here, so consider the importance of phenotype noise. We put some noise here and study the evolution under noise or without noise. And actually the result here is that after many generations and the distribution of fitness is the number of genes. So if so actually we normalize that this is best. So all genes are on after this. So these target genes are all are on. And if the number is off, increases equals like this. And so the evolution under large noise, it has a very sharp distribution. So all mutants or all organisms here is in the highest fittest state. But in a small noise case, so some are of course good, but there are many, many bad ones. And actually, if we just plot this average fitness, and there is some threshold, and this is noise strength. And around here, it suddenly starts to drop. So if the noise is very small, we cannot have a robust evolution. We cannot, so some mutants are always bad. And actually, we check if this value corresponds to the previous relationship, this. And so to check that, so again we plot VIP and VG, and for given course of evolution, this so is again proportionally decreases. If this is so sigma large, so noise is larger, and actually if the noise is larger, it goes like this. But if you decrease the noise, then around this, so there is some threshold value, and this decrease stops, and it cannot go there. And actually, this corresponds to this value of VG equals 3. So again, the previous theoretical argument seems to be all to be satisfied. So, so I checked why this is so, and basically, even for the fittest one, the dynamic structure is quite different. In the case of large noise case, so starting from some gene expression dynamics, it smoothly goes here. So starting from many initial conditions, this very smoothly, robustly goes to this target position. But in the case of low noise case, what we observed here is that starting from given initial condition, it goes here. But if you pattern, then it goes to a different position easily. So the evolved network structure itself has this, in this large noise case, has a high robustness. And in this case, low noise case, is very little, no robustness. And there is some kind of transition here. And the reason can be roughly argued by this. So if you start, so from this, so the task we put here is that starting here and going to go. But the dynamics is quite complicated. So there are many turning points. And from each, if it hit, it may go to a different position. And such turning points are distributed in this space of gene expression dynamics. And then, so if we make evolution under small nodes, they try to make a dynamic such that these turning points are not within this area. This delta is this noise area. So if this original orbit is part of the little bit by noise, but it does not hit this. And so that such dynamics are so selective to this evolution. But in the large noise case, they try to make such dynamics so that this turning point does not hit within this noise allowance range. So far both are fitting. But then if you consider the mutation, by the mutation, this turning point's position can roughly change with some rate. Then it easily touches here, in this case, or it touches the original orbit. But in this case, it may touch here, but it does not hit the original orbit. So the previous structure is preserved. And so that robust evolution is possible. And so probably this is the mechanism why this robust evolution is possible under phenotype noise, under large enough phenotype noise. And so we think that this is quite general in a system such time that fitness is determined after developmental dynamics. And this developmental dynamics need some okay, so I'd like to mention some so discussion of evolution of robustness. And so robustness means that insensitivity of fitness or phenotype to systems change. And there are several means. Robustness against noise during the developmental process, or robustness against parameter change by mutation. And this robustness can be measured by this VIP and VG, because VIP means that variance of phenotype against the noise, the same gene. But VG is that against mutation. So if VIP is large or small, it's more robust. And VG is small, then it has more mutational robustness. And if VIP is small, it has more developmental robustness. So what we observe here, that if this is satisfied, then both start to decrease, VIP and VG decrease. So that means this robustness increases. So in that sense, noise is necessary for evolution of robustness. And also, this result, VIP proportional to VG, is that developmental robustness and mutational or genetic robustness are somewhat linked. And this is nothing but what Waddington tried to say, I think, genetic assimilation of phenotype change. And maybe, so this is an argument that Waddington's argument in terms of the Chateaubriand principle and this VG-VP relationship that I think is. And so, and this casts a kind of new picture on the old question on nature versus nurture. So in the standard population genetics, non-genetic variation are regarded to be environmental variation. And anyway, the ratio of genetic variation to total variation, to total phenotypic variation, is called heritability. And this value, actually, for most cases, is less than 0.5. Actually, for philosophy of this. And our argument is that, so the total phenotype variance, if we can sum independently, that is VIP plus VG. And the one, due to genetic one, is this VG. So that means, in our argument, this is like this. So our inequality means that this should be less than half. Or in other words, this means inequality that nothing is more important than nature. That is in Japanese proverb. But anyway, so at least this is true for phenotype relevance to fitness. But this is a very strong statement, so I'm not sure how much this can be applied to general. But anyway, so far this theoretical result and many simulations seem to satisfy, and we try to make a detailed experimental confirmation. Okay, so maybe I have to skip all this. And there is a story about this adaptation in terms of consistency between cell growth and stochastic gene expression dynamics, and that leads to some kind of general adaptation mechanism without signal transduction. I think I have to skip this. And also, as for the application of this consistency, maybe the most straightforward one is that cell reproduction and multicellular organism. And so we consider some kind of catalytic reaction network of cells. And then considering reproduction of cell is consistent with this multicellular growth condition. Then that leads to some kind of cellular differentiation or stem cell differentiation and some rules for the regulation of the probability of the differentiation. But I think I have to skip this. And so to sum up, so by considering some consistency principle for biology, so first we consider replication of molecules and cells, and this leads to some kind of universal statistical laws, and that has somehow checked in the present cell also. And the second, I consider some kind of genetic and phenotype relationship. And first we saw some kind of phenotypic fluctuation and the evolution speed of these are proportional. And then I discussed the relationship between isogenic phenotype fluctuation versus phenotype variation by mutation. And then, so there is some kind of inequality and proportionality. And we see that robustness to mutation and robustness to developmental noise are tightly linked. And that somehow, so we can have some more mathematical representation or quantitative representation of Waddington's argument about 50 years ago. And also, so I do not have time to go into this, but adaptation of internal cellular state and growth. And then this cell growth system has a kind of general adaptation mechanism. So exploiting noise, even without explicit signal transduction. And the one is that, so differentiation from stem cell and developmental robustness are discussed from this consistency between replication of cells and cell ensembles. And so of course, this consistency is just for basically stable state, stable reproduction, stable evolutionary state. So for example, speciation case, we need some kind of other mechanism. And so how this consistency is broken down and restored, that is necessary. So that is more related to dynamical system studies like analysis. But anyway, so let's hope. Yeah, these are collaborators. Okay, thank you.
Thank you very much. Yes, any questions or comments, please?
Thank you for a very interesting talk. And what strikes me as a biologist is that, in fact, you could adapt this easily to describe a real-life phenomenon. And that is that C57 black isogenic female mice have large differences in the number of the oocytes. This was described in a book that Tuck Finch and Tom Kirkwood wrote a few years ago called Chance, Evolution, and Longevity or something. But the standing explanation is that the females start off with large numbers of oocytes in the liver, and then their cells stop dividing. Part of that difference is when they receive the signal to stop dividing, and that's a stochastic function. And then there's a lag in response time to the signal. You can't stop anytime. And so you can explain by just those two features, and there's a subsequent period of selection, which can also be partial stochastic. But you can then, I think, explain in principle the numbers of how an isogenic female and an iso environment can yield twofold variations in oocyte numbers. And the point of that system is that there's numbers and a lot of descriptive biology. It'd be interesting to see if you could take, granted, a one-cell math model, but applied to multi-site interacting system. I think you probably could. It might be interesting to see if you come out with a reasonable explanation.
Yeah. So in this evolution study, so at least in the model, I consider basically single cell. So,
Yes.
but as for this original distribution theory, maybe this does not depend on which this is a single cell organism or sexual organism. It's just very general.
Yes.
So probably we can apply this to multi-cell organism also. And so that's interesting.
And there are several examples.
Yeah. Yeah.
But are real life examples of biology. So
Yes.
in that book.
Yeah.
You could look at it,
Yeah.
see the data and then see
Yeah.
what you can do.
Yeah. So in some sense, what I want to try is that kind of theory like thermodynamics. So it's general, independent of which molecule is involved. But so kind of from the stability, we can have general theory of this macroscopic system. And so I think that's an ideal theory of it. And so, but also it's important how this is realized through models. So it's like a statistical physics. So combining, so study that direction is also important.
And the second comment is that you can't have the evolution without noise.
Yeah.
And yesterday we heard that our brains don't function well without noise.
Yeah.
And so perhaps there is a consistency among the different students.
Yeah. At least this may please Professor Yanagita.
I have a comment based on the previous question, and then I have a question of my own. The comment is, biologists know that inbred strains which are tending towards isogenicity, so they are not genetically identical, but the genetic variation gets smaller and smaller. Biologists know that inbred strains show much more variation than outbred strains in the phenotype. So this seems to, in a loose way, support the notion that the more genetically similar a group of, let's say, mice, the larger the phenotypic variation. However, they also know that if you have an F1 cross hybrid between two inbred strains, the progeny of that cross are also genetically very similar. In fact, in principle, they are identical. But they show very little phenotypic variation. In fact, the way to minimize phenotypic variation, and geneticists do this routinely in laboratory experiments, is to work not with inbred strains, but with the progeny of two inbred strains, so that you have heterozygotes at a great many loci. The individuals are genetically identical or close to being genetically identical, but you have suppressed phenotypic variation. So there is a kind of discordance between genetic variation and phenotypic variation. That's a comment which I put forward for you. The question has to do both with your experimental observation and with the network model which you use to validate that observation.
Yeah.
So the observation had to do with selecting for increased fluorescence in these so-called isogenic populations of bacteria. My question is the following. At any given time you work with large numbers of bacteria. I presume that you check that there is no genetic change at the locus of interest to you, which is the promoter in front of the fluorescence gene. But how are you sure that there is no genetic variation at a great many other loci which you are not looking at because you think they are uninteresting? Now those variations may very well be contributing to phenotypic variations at the locus which is of interest to you. And in fact, in the theoretical model where you pick the top 5%, as you put it, after mutating the network randomly, surely that top 5% consists of genetically heterogeneous cells. So my question is, are you really dealing with a situation of genetic identity here at all, whether in the experiment or in the model?
Okay, so in this model, at least in that model, so we take this phenotype fluctuation, for example, when we measure phenotype fluctuation, we choose the same network. So we have many networks, different networks, and for this network, this thickness is very large, and for this second network, for this And so we set it here for the next generation. But when we measure VP, we take this and measure this phenotype fluctuation. So for this case, so since this is simulation, we are sure that it comes from the same gene. And in the experiment, of course, it's difficult. And actually, in this experiment, so when we put mutations, so here, when we make mutagenesis, so for genes that are responsible for these fluorescent proteins, we make a high mutation rate, put this by this mutation, mutagenesis. For this, very high. And for some others, so there can be so small mutation per generation. So it's possible, but maybe this can be regarded as noise or something like that.
No, my point was simply the following. What you call the clone,
Yeah.
is it really a clone?
Yeah. So in this experiment, so I think that probably, at least in this experiment, so there may be a possibility that there are some small mutation or change in other parts. But I don't think that is not so much responsible for this. Or at least there may be some, different ones. But when we measure, for example, this distribution here, maybe some mutants with different part may be included, but I guess that fraction is not so much to change drastically this big. But that's, we need to check. Yeah.
Thank you for very nice, stimulating talk. What I have shown there is almost similar subject, so the evolutional process.
Yeah.
My question is, we usually use fitness for evolutional processes, but fitness is a kind of abstract concept, which is not in real space.
Yeah.
So how do you think meaning of the fitness?
Yeah.
I always wonder what is
So-
the fitness.
So basically, so how it grows, so and divide, and so just growth speed. And we can just consider the growth speed of organism. And so, but for example, in the nature, in this case, so this growth speed is depending on distribution of other organisms. And so fitness is not defined as a quantity for this single organism, but depending on the distribution of other organisms. So, for example, to consider speciation, I think that this is quite important, and I discussed this a few years ago. But so instead of going into such questions, here we consider only artificial selection process. So in this case, so from the outside we put this condition for this fitness. And so in this case, so fitness as a growth speed is determined from our side. And for example, when Darwin started his evolution theory, he considered a lot about this evolution of cattle, so it's an artificial selection process, and he mostly uses the results from there. So in that case, to start from this evolution and phenotype fluctuation relationship, in the beginning, it's good to start from this simple situation with given fitness given as from the outside as artificial selection. Thank you.
Okay, it's time to have a break. Thank you. Sorry about